Firstly, let's look at the testis. The testis is surrounded by a capsule which consists of tunica vaginalis, tunica aborigina, and tunica vasculosa. Tunica aborigina is a thick, dense connective tissue which is thickened on the posterior surface of the testis to form the mediastinum testis, from which fibrous septa penetrate the gland, dividing it into about 250 pyramidal compartments called testicular lobules. Each testicular lobule is occupied by 1 to 4 seminiferous tubules, enmeshed in a web of loose connective tissue, lattice rich in blood and lymphatic vessels, nerves, and interstitial cells of all Leydig cells. Seminiferous tubules produce male reproductive cells, the spermatozoa. Leydig cells secrete testicular androgens. We will focus on seminiferous tubule, Leydig cell, and blood testis barrier. This is a light micrograph of the testis stained by HE. Star shows the tunica aborigina. The single arrow indicates one septum. Square shows the tunica vasculosa. Double arrows indicate the seminiferous tubule. Let's look at the seminiferous tubule. Each testis has 250 to 1,000 seminiferous tubules, which are 30 to 70 centimeters long. The seminiferous tubule is lined with complex stratified epithelium called spermatogenic epithelium, which consists of spermatogenic cells and Sertoli cells, all supporting cells, based on the basement membrane. This is the pattern diagram of the cross-section of the seminiferous tubule. The arrow indicates the basement membrane. The star shows the lumen of the tubule. The wall of the tubule is the spermatogenic epithelium. This is the light micrograph of the seminiferous tubule. Spermatogenic cells are arranged in 4 to 8 layers from the basal of the seminiferous tubule to lumen as they mature. They are spermatogonium, primary spermatocyte, secondary spermatocyte, spermatid, and spermatozoon. Spermatogonia successively divide and eventually differentiate into spermatozoa. This process is called spermatogenesis. In this pattern diagram, you can see the spermatogonium, primary spermatocyte, secondary spermatocyte, spermatid, and spermatozoon from the basal to the lumen. The process begins with a primitive germ cell called spermatogonium. Spermatogonium is a relatively small round or ellipsoid cell, about 12 micrometers in diameter, situated adjacent to basal membrane. Pale staining cytoplasm. Nuclei are round or ovoid. Double arrows indicate the spermatogonium. Primary spermatocyte. The spermatogonium diploid cell duplicate their DNA to become tetraploid cell. Primary spermatocyte. They are round cells, become the largest, about 18 micrometers in diameter, the left basal layer. And the nucleus is large, round, and on different stage of division. Through first meiosis, primary spermatocyte divides into two secondary spermatocytes. In this picture, the arrow indicates the primary spermatocyte. Secondary spermatocyte. They are round cells, 12 micrometers in diameter. Nucleus is round, deep, stained. Secondary spermatocytes are difficult to observe in section because they are short-lived cells that remain in interface very briefly and quickly enter into the second meiotic division. Division of each secondary spermatocyte results in 2 cells that contain 23 chromosomes called spermatids. In this picture, the arrow indicates the secondary spermatocyte. Spermatid. They are spherical or polygonal. They are the smallest cells, 8 micrometers in diameter, lying close to the lumen. Spermatids are haploid not only in terms of number of chromosomes, but also in the amount of DNA. No further division occurs in spermatids. In this light micrograph, S means spermatids. Spermatozoon. They are 60 micrometers in length. The head is pear-shaped and flattened, containing nucleus and acrosome. The acrosome is specific lysosome covering the anterior 2/3 of the nucleus. The tail is the flagellum, derived from the centriole. Tail is subdivided from end to post into 4 segments: neck, middle segment, principal segment, and end segment. In this picture, the arrow indicates the spermatozoon. This is the sperm smear. You can see the head of the spermatozoon. The square shows. The arrow indicates the acrosome, pale stained. Spermiogenesis is the process from the spermatids to spermatozoa. It includes formation of the acrosome covering the anterior and lateral portion of the nucleus, containing hydrolytic enzymes for fertilization, condensation and elongation of nucleus, formation of flagellum. Mitochondria aggregate around the proximal part of the flagellum, forming mitochondrial sheath. Discharge of useless organelles and cytoplasm, also named residual body. In humans, the changes that occur between the spermatogonial state and the formation of the spermatozoa take about 64 days. Spermatogenesis must occur at a temperature of 2 to 3 degrees centigrade lower than the abdominal temperature. During the process of spermatogenesis and spermiogenesis, some malformed sperm are usually formed. Malformed sperm accounts for 20% to 40% in the semen of male with normal fertility. If the number is above 40%, infertility can be caused. These are normal sperm. Sertoli cells are also called supporting cells, which are important for the function of the testis. Sertoli cells are columnar or pyramidal cells with elongated nucleus. Nucleus is triangular, ovoid, and pale stained with prominent nucleolus. The cells rest on the basal lamina. Their apical ends extend into lumen of seminiferous tubule. The outlines of certain cells appear poorly defined because of the numerous lateral processes that surround spermatogenic cells. Ultrastructure. Certain cells contain abundant SER, some RER, well-developed Golgi complex, numerous mitochondria, lysosomes, glycogens, and lipid droplets. Adjacent certain cells are bound together by tight junctions at the basolateral part of the cells. Certain cells have several functions: support, protect, nourish, regulate and release germinal cells, secrete androgen binding protein, which can bind to androgen, maintain the level of androgen concentration of lumen, and help the spermatogenesis. Phagocytosis, the generated cells and spermogenic residual bodies, constitute the blood-tested barrier. Blood-tested barrier is between the blood and the interior of the seminiferous tubules, including endothelium, basal lamina of endothelium, connective tissue, basal lamina of seminiferous tubule, and tight junction of certain cell. Blood-tested barrier can be resistant to most harmful factors, protect the seminiferous cells from autoimmune reaction. Interstitial tissue. The interstitial tissue of the testis is an important site of production of androgens. Loose connective tissue extends from tunica vesiculosa and fills spaces between seminiferous tubules. Leydig cells are located in the connective tissue. They are endocrine cells in groups. Leydig cells are large polygonal-shaped cells with round nucleus. The cytoplasm is acidophilic. Leydig cells have features of steroid hormone-secreting cells. Main function is to secrete testosterone androgen. Okay, above is the content of the testis.